Yeah, thank you very much. Thank you, Mohammed. And yeah, thank you for the introduction and also the opportunity to share some of our work here. It's really a pleasure to be here. So, like Mohammed said, I will just share some of our work that we are doing on the effect of the tumor microenvironment on cancer cell metabolism. I will just start with a quick introduction and some of my background. So, I did my master's degree in South Africa at the Northwest University. I then went to the Netherlands, to the University of Utrecht, where I did my PhD in metabolomics and aging. And then after my PhD, I went to Glasgow in Scotland to the Beaton Cancer Research Institute, where I started to study cancer metabolism. And my postdoc, the whole lab, then we moved to London to the Francis Crick Institute, where I finalized my postdoc in cancer metabolism. And then I had a 3-year stint in industry where I worked for a company in Cambridge in the UK called Elstone Medical, where we developed some diagnostic tests for lung cancer. And since the last 2 years now, I am back in South Africa to my university, my alma mater, where I am now a senior lecturer at the biochemistry department. And here you can see Potchefstroom, where the Northwest University is just a bit southwest of Johannesburg. So yeah, in our research group, we are interested in cancer metabolism, and in the last 30 years or so, there's been a lot of advancement in understanding the metabolism of the primary tumor, how cancer cells adapt their metabolism to support proliferation, etc. But there's less known about the metabolic changes cancer cells undergo during metastasis, during circulation, when the circulating tumor cells are in circulation. How do they survive that? How do they adapt their metabolism? And also for recolonization, then the cells have to switch again back their proliferation, reprogram again to reproliferate again, to form secondary metastatic sites. So, and also we are very interested to see the effect of the tumor microenvironment. For instance, we all know that solid tumors grow very rapidly, and because of the disorganization of the vasculature and also the distance, the further the tumor grows away from the blood supply, we get a gradient of oxygen, nutrients, and pH. So in our group, we became very interested to see what is the effect of pH, changes in pH on the cell metabolism. So like I already said, we already know, we've all probably heard about the Warburg effect, which states that even in sufficient levels of oxygen, tumor cells prefer to use glucose instead of or to use glycolysis and produce lactate instead of using mitochondrial respiration. And the hypothesis behind this is that these tumor cells need to generate building blocks to support these high proliferation rates that they have. And through glycolysis, it can shunt this pathway into various anabolic pathways, whereas normal cells use most of their glucose to oxidize it in mitochondrial respiration for energy production. But like I said, there's not that much known about what the cells, how the cells adapt their metabolism during metastasis. So to shed some more light on this, we used two very simple models, in vitro cell culture models. So the first one is we just take cancer cell lines and we grow them at different pHs. So we we take the cell lines and we then just seed them into flask, and we adjust the pH of the media. We use specific buffers, etc. We make the media from powder, and then we use specific buffers to adjust the pH. And then we just grow these cells for a minimum of 3 days in the low pH, but we also grow them for longer periods, up to 1 month. And then we do some metabolic characterization and also some proliferation assays, migration, etc. And this represents this, this pH gradient that we see in the primary tumor. And then to mimic in a very simple way the metastatic cascade, we take cells that grow in normal 2D in attached to cell culture plates. We then grow them in ultra low adherent plates, so these cells cannot adhere to the surface, so they stay in suspension. And this represents, in a simple way, the cells in circulation. And then we after a period of time that they grow in circulation, we reintroduce them to adherent plates where they are able to reattach and basically recolonize. And then in our model representing the formation of primary metastatic sites. So this is, we realize this is a very simple model, but we do think that we can get some information to some extent on the metabolic adaptations these cancer cells have to undergo to survive or to manage to go through this whole process. So first of all, I will just show some data we collected on the effect of the pH on the cells, and then lastly, I have one slide on the metastatic cascade. So first of all, we looked at the proliferation of the cells grown at the different pH levels, and we grew them for 3 days and counted the cells every day. And you can see that the physiological pH, the cells grew at a normal rate, but at the lower pHs, there was definitely inhibition of cell proliferation. So this was not that surprising, because there are some data already in the literature showing that the pH can affect the proliferation of the cells. So next, we were interested in the ability of these cells to adhere, because we noticed that at the lower pH, the cells were not that, they didn't adhere to the culture plate that easily. So we did a sort of adherence assay where we seeded exactly the same amount of cells for each pH condition. And then after a day, we just collected the media from those wells, and we counted the live cells in the media. So we can see here that in the low pH, the pH 6.4, there was a lot of cells in the media, just indicating that they didn't adhere to the culture plate. Where in the higher pHs, especially in the physiological pH, almost all of the cells, adhered in that period of time. So it shows that there's a lack of adherence, ability to adhere in the low pH cells. And we then did also a migration assay using the transwell assay, where we seeded the cells in the top chamber and allowed the cells to migrate for a certain period of time, and then measured the cells that migrated. And we could see that in the low pH 6.4, there was significantly more cells that migrated through the membrane. And this all pointed towards a sort of phenotype that resembles epithelial to mesenchymal transition, so the EMT. So we decided to look at some EMT markers, and indeed we could see that there was a reduction in the expression of E-cadherin, which is a epithelial marker, and is a a molecule that helps the cells to cell adherence and also cell surface adherence. So that makes sense that there was a lack of adherence in the low pH cells. And also we saw this Western blot's not perfect, but we saw a hint of increase in vimentin, which is a mesenchymal marker. So it looks like there's some sort of EMT happening in the cells growing at a lower pH, and that would also explain the reduced proliferation and the increased in migration capabilities. So we then asked, how does this switch EMT, how does it affect the metabolism of these cells? So the first thing we did, we looked at the respiration, mitochondrial respiration of these cells, and we used the Seahorse analyzer to measure oxygen consumption rates in the different conditions. And we were quite surprised to see that the cells grown at a low pH, the lowest pH, had the highest oxygen consumption rate and also had the highest maximum respiration capacity, as you can see here, meaning that they consume more oxygen compared to the other conditions, and it seems to be through mitochondrial respiration. We then looked at the amount of mitochondria in the cells using the fluorescent probe MitoTracker Green, which binds to mitochondria, and we saw a significant increase in the mitochondrial mass in these cells growing at a lower pH. So that is also in line with the increased respiration. So we then decided to look a bit closer to the metabolism, and we used a technique called stable isotope tracing. In this technique, we can give cells in cell culture, we give them stable isotope labeled nutrients like glucose. So you can see in the blue dots representing 13C carbons, and the white dots representing normal 12C carbons. So this is normal glucose, but then we give the cells this 13C labeled glucose. So all 6 carbons of the glucose is labeled with this stable isotope. So this makes the glucose 6 daltons heavier than normal glucose, and we can then distinguish it from normal glucose in the mass spectrometer. So we then use LCMS to measure it. So we give this labeled glucose to the cells in culture, and then we incubate them for 6 hours with the glucose, and then we subject the cells to LCMS. So we extract the intracellular metabolites, and we perform then LCMS analysis. So we can then track the glucose into the different pathways. We can see if they use it for in the TCA cycle, or do they produce lactate, or do they shunt it into the pentose phosphate pathway, etc. So we did this for the different pH conditions, and we saw, interestingly, that there was much less glucose in the intracellular glucose in the lower pH cells. We haven't yet established if it's because they use it quicker or if they take up less. But if we look at the lactate levels here, intracellular lactate, we see that there's an increase in the lactate production at the lower pH conditions, and that there's a decrease of glucose being used in the TCA cycle. So much less glucose is being used for respiration or to produce, to be oxidized, that can be used for oxphos. So to some extent, it makes sense, but also, yeah, we didn't expect to see an increase in lactate production when there's an increase in respiration. So this was a bit of a puzzle to us. But next, we try to look at glutamine metabolism. So glutamine is quite heavily utilized by cancer cells and is shunted directly into the TCA cycle through glutamate and alpha ketoglutarate. So we decided to give the cells a stable isotope labeled glutamine. So we could see the glutamine got converted to glutamate and then eventually to citrate, after going through the TCA cycle. And again, we were quite surprised to see that the cells at the lower pH used much less glutamine in the TCA cycle. And the TCA, combining this data with the glucose label data, we could see that the TCA cycle is running very slowly, which is surprising taking into account the high respiration rates. And we also saw a reduction in glutathione, just indicating that these cells are under some sort of stress, because there's if you look at the oxidized to reduced glutathione ratio, we can see that there is some oxidative stress in these cells at the lower pH. So we're still not quite sure what's going on here with the metabolism, where we see an increased mitochondrial respiration, but we see a decrease in the TCA cycle activity. So one reason could be that these cells use fatty acid oxidation to generate reducing agents like NADH and FADH, and directly shuttle this to the electron transport chain, or the OXPHOS, and so bypassing the TCA cycle. So this prompted us to, this just shows that there was an increase in glutamine consumption. So the cells at the low pH do use the glutamine, but we're not sure how yet. So we're still trying to figure that out. But it prompted us to look at lipid metabolism. So first of all, we looked at lipid accumulation in these cells. And we saw that at the lower pH, there's a massive increase in lipid droplet formation. So these cells do store lipids, at a much higher capacity than the other cells. And actually, you can even see this through the microscope in normal phase contrast microscopy. You could see the increased lipid droplets in these cells compared to the physiological pH. We then did a lipidomics analysis on the mass spectrometer, and we could see at the low pH conditions, the pH 6.4 and 6.8, there's a huge increase in these lipid species that elute here in this area of the column. And these lipid species correlate to triacylglycerols and diacylglycerols, which indeed is the sort of lipids that gets stored by the cells in the lipid droplets. We also look at DGAT expression. So DGAT is the diacylglycerol acyl transferase, which is the enzyme that catalyzes the last step in the triacylglyceride formation, and then these lipids are stored in the lipid droplets. So we saw increasing DGAT expression at the lower pH, and when we inhibit DGAT with a DGAT inhibitor, we indeed see a disappearance of these lipid species eluting here, and there was also almost no lipid droplets in these cells. And then we looked at the cell viability when we inhibit the lipid droplet formation, and we saw a significant reduction in cell viability at the low pH. And this indicates to us that this lipid droplets do play an important role in these cells at the low pH. So we are now busy with experiments to determine if the cells use these lipids for fatty acid oxidation to produce energy. We also know that these cells at the lower pH requires quite a lot of ATP to maintain the neutral intracellular pH, because they grow in this acidic environment, but the intracellular pH needs to be physiological, and for that there's a lot of ATPase dependent proton pumps that have to work hard to keep this intracellular pH neutral. So we're now busy with experiments to see if the cells uses this lipids for energy production. And then just quickly, I would like to touch on the metastasis cascade model, where we grow the cells in normal 2D conditions, and then we grow them. We seed them in the ultra low adherence plates, where they then grow in suspension for up to 12 days, and then we resuspend them reculture them in the normal adherence plates, and you can see that the cells then reattach and they start to proliferate again and start to migrate towards the space. And interestingly, you can see there's some morphological changes in the cells, and we haven't quite yet figured out what these are, but we think it's the cells that are in the middle of the in the center of these cell clusters that have some morphological changes, and it's likely that they are also a more mesenchymal phenotype. But we also did some metabolic studies, especially mitochondrial metabolism, and we saw an increase in mitochondrial ROS production when the cells are in suspension growing as detached. And if we put them back into reattached conditions, this mitochondrial ROS levels decreased again. And we also saw the mitochondrial membrane potential decrease in the detached condition, and then upon reattachment it recovers again. So, and again, the cells growing in detached conditions had a very, very low oxygen consumption rate. So I think what this data highlights is how metabolically plastic these cancer cells need to be to go through all these processes, or being exposed to all these conditions, low pH conditions. Then they have to grow in suspension, they have to survive anoicus, all these stress that they experience during suspension, and then upon recolonization they have to be able to switch back to a proliferative state, and switch back their metabolism in order to support proliferation, etc. So this is just some very preliminary data. We are doing these sort of experiments using these cell culture models in our lab, and we are still ongoing with this work, but I'm happy to share some of these data with you. And then lastly, I would just like to thank my two students, Jaco Gerber and Ranay Lindeke, who were also at the SAS-BMB conference where we met Mohammed, and they are responsible for this work, and I would like to thank you for your attention. And if there's any questions, I'll be happy to answer.
Thank you very much, Stan. Thank you very much. So we have room for only 2 questions because they are quite to kick us from the amphitheater. So room for 2 questions. Yes.
So first question is, what about the normal cell, not tumoral cell?
Yeah.
What is the effect of variable pH? Then how to explain the lipid droplets support cell survival at low pH? Why? Is it some resistance to immunoresponse or camouflage or something like this? And why in your profile it's not the same cell number when you compare the 3
the first slide, you mean? Yes. So let's summarize the question. So it will be the same thing, you think, Stan, if we deal with primary cell or normal cell, I mean non-tumorigenic cells, regarding the when we acidify the medium. So when we have low pH, so 5 minutes maximum. Okay. So I will just keep this question because we have only 5 minutes. So what happened? What do you think when we have low pH in normal cells?
Yeah, that's a very good question, and to be honest, I don't know the answer to that. But it would be interesting to see if normal cells would be able to adapt in a similar way. You do get some acidosis during inflammation, etc. So, it could be interesting to look at that.
Okay, okay.
I have a question if possible.
So for the quiz, Stan, thank you very much because they
Yeah.
are trying to
Okay.
kick us from the office.
Okay, no problem. Thank you so
Thank you
much.
very much. I will write to you. I will write to you.
Okay, thank you. Thanks, everyone.
So for the quiz, Mariam, tu peux partager? Yes. So the quiz, we will keep the quiz for tomorrow, Mariam, and we will try to do it like we will try to do 3 quiz for tomorrow. Okay, thank you very much. Merci beaucoup.
Thank you, Dr. Mohammed, for this
شكراً
great
حبيبي.
work.
شكرا.